Welcome to this presentation. I'm going to talk about nutritional epidemiology. And in this session, we are going to look at the definition of nutritional epidemiology, the objectives, nutritional epidemiology, and then the approaches. And then finally, we'll look at the dietary assessment methods. So nutritional epidemiology is the study of the relationship between diet and health outcomes in populations. The key terms here are about three, that is health outcomes, and then diet, and then population. So we are not looking at individuals, but the focus is on populations. So we are looking at how the food that people eat influences their health. Okay, so their diet, how does the diet influence health outcomes of populations? Okay, so we understand already that diet is closely related to the occurrence of diseases. So, for example, high salt foods increase the risk for hypertension. Sugary foods are associated with an increase in diabetes type 2. So there is an association already between diet and the occurrence of disease conditions. So we want to basically understand how diet results into diseases, how diet can actually enable one to have better health outcomes or stay healthy in their lives. So that is basically what we look at in epidemiology, in nutritional epidemiology. So what we basically look at, just a minute, I think getting just 1 minute. Okay, so we understand that conditions are associated, we have conditions that are associated with diet, and if we understand them and what dietary factors that are resulting to them, then we can be able to institute control measures in terms of policies, in terms of fortification, in terms of interventions, a number of interventions that we can actually put in place. So we basically, throughout this presentation, we shall be talking about certain concepts that you should be aware about. The first concept is what we call exposures. Okay, so when people eat, okay, when they eat certain foods, or when they eat food, we are saying that the specific foods that they have eaten may put them at a certain risk. And that is what we are calling as exposures. So we are basically looking at the dietary factors. What dietary factors, or what diets are people eating, and they are putting them at what risk. So those risk factors in terms of dietary factors are the ones we are calling the exposures. And then we come to the next concept, which is outcomes. So when you eat a particular food, it may put you at a particular risk, or at a risk of getting a particular condition. So that particular condition is what we are calling a health outcome. So it can be that the health outcome can be acquiring a new disease, okay, or having complications associated with a particular risk factor. So outcomes just basically means what is the result of you taking certain foods. Okay, that is what you call outcomes. And then the other concept that we should be aware about that you had earlier on explained is that our focus is on populations. These populations can be schools, they can be communities, they can be a group of individuals, let us say athletics, okay, a number of individuals. They're the ones that we are focusing on. We are not focusing on individuals, but we are focusing on groups of individuals who could be connected by perhaps geographical location or another pattern, such as a social setting, such as a church, such as a school. Okay, so we focus on populations rather than individuals. So the interest of nutrition epidemiology is basically to look at the foods that people eat, what health outcomes are associated with them. Okay, so we want to identify that kind of association. And for the different foods, how do they result into these health outcomes? They are basically interested in understanding the mechanisms through which these foods actually result into different health outcomes. So remember, health outcomes are either acquiring a certain disease, it can be hypertension, it can be diabetes, it can be increasing a certain risk factor, let us say cholesterol. Those are all health outcomes. And then we are also interested in getting knowledge that we can use to inform the interventions or the policy or the guidelines that we can put in place to safeguard the population. So we basically use knowledge of epidemiology in informing policies, designing programs, in also educating people about different healthy eating habits. We also use knowledge from epidemiology in informing decisions about guidelines, policies, and then also what we call dietary guidelines. So we use nutrition epidemiology in a number of fields. So epidemiology as a field is quite challenging because of a number of factors. One, we need to understand what people are actually eating for us to know whether there are dietary patterns associated with particular health outcomes. So sometimes measuring this comes with its own difficulties. It is not always very easy to really understand what someone has eaten for the last, let us say, 1 month, 2 weeks, 1 week. It's not very easy to measure. So we have measurement problems. So that is one major problem of nutrition epidemiology. The second problem is what we call confounding factors. Confounding factors are those factors that actually can influence the outcome of interest. So you may think that people who eat vegetables are likely to stay healthy or do not have reduced risks for getting, let us say, hypertension. But you may realize that actually those people may have another characteristic that may also influence the likelihood of getting hypertension. Maybe the person is eating vegetables, yes, but that person still is exercising. So that means that if you're investigating vegetables with hypertension, when you neglect that the person might actually be exercising, and that exercising is also contributing to the reduced risk for hypertension, then you're not doing something right. So it means there is another factor that is actually influencing the outcome of interest, and we should take care of it. Okay, so if it is very important that you establish the fact that there could be other confounding factors that you really need to take care of. Okay, so we move to the next slide. So the next slide is about the fact that for a given disease to happen, for example, for a person to get, let us say, hypertension, there could be several factors that are actually interacting. So you cannot basically say because of only taking foods with high salt content. It could be a genetic factor. It could be an issue to do with the environmental situation, or it could be a social problem that is associated with the health outcome of interest. So isolating these health outcomes with their risk factors is very hard, and therefore it is one of the challenges of nutritional epidemiology. Okay, so now let's move to one key aspect of nutritional epidemiology. We already said that we need to understand what people are eating and also link what people are eating to the diseases that are happening within the population. So the first thing is to do is to assess what are they eating. So to do that, we carry out what we call dietary assessments. So with dietary assessments, we can get data that we can use to relate to what health conditions are happening within the population. So we have a variety of ways in which we can do these assessments. We have the 24-hour dietary report, the food frequency questionnaires, the food records and diaries, and then biomarkers and other objective measures. We can use those methods. So let's look at each of those. So 24-hour report, just like the name suggests, we ask individuals to write down all the foods and beverages that they have consumed in the last 24 hours. Okay, so it means that the person needs to have a very good memory, such that they tell us what they have eaten and how much, that is proportion. So what varieties have you eaten and what proportions? The advantage of this is that actually you have a lot of information that you're going to get that is actually detailed about the different foods that someone has actually eaten and how much. So it is not really troublesome. It's relatively easy to actually carry out. But the challenge is that some people may not actually have good memories to be able to tell you every food that they have actually eaten. Consequently, there could be issues with accuracy of what they are actually telling you. So this brings issues to do with recall bias. So if you ask people about what they have eaten in the last 24 hours, it may not be a very good indicator for how they have been eating for the last 1 week. It could be that today I've just decided to fast, and then I tell you that I've just been taking just plain water. So that means that you will not get the real pattern of my eating just because that today I've just decided to change my diet. So the other method, which is the food frequency questionnaire, with this, we are assessing the frequency and the proportion or the portions of those, the portion sizes of selected foods over a specific period of time. So you basically look at the major foods that the person is likely to eat, and then look at how much they are eating, and then how frequent are they eating these foods. And then, of course, you tabulate this data and understand their overall eating patterns or dietary patterns. It is very easy to do this, and it is also cost effective, but still it relies on the person's ability to recall. Apportioning the sizes can be quite challenging and may be basically imprecise. And definitely it's not like the 24-hour recall. So it may not really capture the day-to-day variations in what someone is actually eating. You can decide to use what we call food records and diaries. With this, the person who is who you are actually studying, just 1 minute. Okay, so the participants or the individuals that you are actually interested in can record their food and beverages that they are actually consuming in real time. So, for example, if I'm going to take a soda, I just note that I'm taking a soda, 300 mils. Okay, so it actually provides you with good detailed data. That is actually, it is actually accurate because it is done written in real time. Okay, but of course you're making the participants have a little bit of trouble having to write each and everything that they have eaten or they're going to eat. But the other challenge is also that the participants may not really report some foods that are not socially desirable. Okay, so if I've taken a foods that I know the nutritionist will not be happy about, then I will not record it. Okay, so that's one of the challenge because I want to conform to what the society expects me of. So that concept is what we call social desirability bias, that people fail to tell you the information that they have because of the fact that that information may be against the social norms or the social expectations. So that creates a kind of bias that we call social desirability bias. And then the last method of assessment is what we are terming as the biomarkers. Okay, the biomarkers is when we actually measure certain, let us say, biomolecules. It could be nutrient levels in our blood or certain minerals in our urine or even still blood. And then this gives us a comprehensive understanding of the levels of particular nutrients in your blood or in your body. So it is actually objective because you are the one who actually takes the sample, and then you you take the measurements. So it is very objective, it is very accurate. So therefore, it does not rely on what people tell you. So the bias problems are actually minimized in this case. The challenge is that you don't want to be pricking people every now and then. Okay, prick me and then get my blood sample. That is invasive and also expensive. You need a laboratory technician. You need also a lab, a working lab. So that is actually something difficult and also cumbersome to do at the operation level. Also, the other problem could be that interpretation of biomarkers could be a challenge. So it brings a lot of issues because other factors also could influence the biomarkers that you're actually investigating and do not necessarily be nutritional issues. So the other thing that is also very important is having looked at all these factors, all these methods of assessments, you need to decide which one you're going to use. So what is your interest? What are you investigating? That will help you in determining what approach you're going to use. So you will look at problems that are associated with these dietary assessments, the issues of bias, issues of underreporting and misreporting, and then also potential social, cultural, and psychological factors that may influence dietary reporting. And also the people who you are going to use to record this data need to actually be well trained. They need to be well trained such that they minimize issues to do with measurement errors and also problems associated with apportioning the different sizes that could bring errors in your measurements. So in practice, it is important that you use a combination of methods. When you do that, it means that you are limiting, basically you're having the strength and weaknesses of each method, but then when we combine, let us say, two methods, it means that you're actually supplementing or reducing the weaknesses of the other, and then while using the strength of the other method. So it could be better that you don't use only one method, but you actually use a combination of them. So we move to the next slide, which is that select the method based on what you're interested in. Basically, these are basically researches. So you have the research design, the objectives, and you have a well-documented plan on how you're going to collect this data. Okay, so it depends basically on what your interest is. Look at the different methods that are available to you, and then see what is the best to use. So do you have any questions? Any questions? Okay, if there are no questions, let's end there, and then let us pick it from there after 10 minutes for the second session.